So continue on with internal defenses. Let's look at cell responses. So one is phagocytosis. So we see this phagocytosis here. So this is where a cell engulfs another cell. This is done by macrophages and monocytes, neutrophils, eosinophils, and mast cells. So, this is just showing that, we pull that microbe into a vesicle, fuses with the lysosome contains digestive enzymes, breaks that organism down. So if we look at this next picture, we can see phagocytosis occurring there as well. Next is chemotaxis. Chemotaxis is where white blood cells are going to move towards and attract chemicals. So here some attracting inflammatory chemicals are being released, and that's going to cause chemotaxis, white blood cells moving towards us. They do this by amoeboid movement, so they push out one part of their body and then they pull the other part forward. All right. So in terms of chemicals, kinins, prostaglandins, leukotrienes, histamine, and complement proteins are all chemicals that cause chemotaxis. Next is diapedesis. So diapedesis is what you see here and actually there as well. So diapedesis is a movement of white blood cells out of a blood vessel into surrounding tissues. So getting them to move out of the blood vessel into the surrounding tissues. It is actually a form of chemotaxis because those chemicals are causing that movement. If we look at particular cells here, one are neutrophils, which just about everything you see here is a neutrophil. Neutrophils are first to infection, and that's because they're the most numerous white blood cell. They're going to recruit and activate other immune cells. The thing about these guys, they're going to die after a single phagocytic event. And then what they do is they release lysosomal enzymes. They're going to kill microorganisms, and then they're going to cause tissue damage and inflammation. So, so this guy here, he gets here. Why he does this is if he kills that microbe there and then he releases a lysosomal enzymes here, that's going to kill more bacteria in the area. So, in doing so, we don't have a whole lot more phagocytosis occurring there. So, kills off a lot of stuff. It's going to damage our tissue as well, but that's okay. We can repair that after we've gotten rid of this infection. So what this creates though is pus. That's the accumulation of dead white blood cells, dead microorganisms, damaged tissue and fluid. Pus looks a little whitish because of the white blood cells. It looks a little greenish because of those damaged tissue and microorganisms here. Next are macrophages, so showing a macrophage here. These are large phagocytic cells. So they're larger than the neutrophils. They're around later in infection because those neutrophils start to die in that area. Macrophages are found throughout our body. Next are basophils and mast cells. You see those guys there. These are release chemicals that cause inflammation. So basophils are in the blood. Mast cells are in our connective tissue. Next are eosinophils. These are going to release chemicals that reduce inflammation. Next are natural killer cells, or NK cells. These are cells that can lyse or burst and kill cancer cells and virus-infected cells. They are a type of lymphocyte. They're going to release perforins. Once again, perforins is another one of these chemicals that creates a hole in the plasma membrane of a pathogen. They also release granzymes. These are going to destroy cellular enzymes and induce apoptosis, so it causes a cell to kill itself. So that's programmed cell death. All right. Next is inflammation. Inflammation is a dilation of blood vessels and accumulation of fluid in an affected organ. So this is a response to physical trauma or a bacterial infection. So what you get from this is vasodilation. So you get more blood to the area, so you get more phagocytic cells there. That's why we want that to occur. It also is going to cause chemotaxis of phagocytic cells to that area. You get increased vascular permeability, so this allows these cells to get into those tissues, and it also allows fibrinogen and complement proteins to enter into the tissue. All right. So what happens here is that fibrinogen is converted into fibrin, and fibrin is a sticky protein, and so what this does is essentially like walls off this area from that infection being able to spread. All right. Next is fever. Fever is an elevation in body temperature above normal. It's a systemic response to invading pathogens. All right, so what by temperature going up, this promotes interferon activation, so those chemicals that interfere with viral replication. It also elevates our metabolic rate and accelerates tissue repair. It also causes our liver and spleen to sequester and store iron and zinc. So sequester means store. So iron and zinc are necessary for microbial growth, so bacterial, fungal, and viral growth. So by getting that out of our system, it's going to help reduce those growth of those pathogens.